Welcome to Antibody Basics by Biointron, Part 12. In this episode, we'll cover antibody formats, specifically multispecific antibodies. Multispecific antibodies are engineered proteins that can recognize and bind to two or more distinct antigens simultaneously. This unique property allows them to bridge interactions between different molecules, activate multiple therapeutic pathways, and enhance target specificity and efficacy. The increasing variability in multispecific formats is a valuable source of diversity that can be applied to the development of biologics for various indications. There are several advantages of using multispecific antibodies, including improved specificity, since they can target multiple antigens on the same cell or bridge interactions between different cell types, improved efficacy as they can activate multiple therapeutic pathways simultaneously, leading to a more potent effect, and multifunctionality, as they can combine antigen binding with other functionalities like immune cell activation or toxin delivery. Currently, multivalent bispecific IgG modified formats predominate out of the over 140 different molecules in clinical testing. However, trispecific and even tetraspecific antibodies are quickly becoming a reality. Here, we'll highlight just a few platforms for tetraspecific antibody production. CrossMab relies on VH or VL or CH1 or CL domain crossover technology in one Fab arm of a bispecific IgG to promote correct assembling of light chains. Knobs into holes technology is also used for heterodimerization of heavy chains. For example, a 4-in-1 crossmab targeting EGFR, HER2, HER3, and VEGF showed superior anti-tumor activity, high avidity binding, and effectiveness at lower concentrations. The Match4 platform involves a core of two split variable domain pairs, each chain containing either two VL or two VH domains, to drive heterodimerization of both chains. Additional single-chain variable fragments appended to the N-terminal end of both chains provide the two extra specificities. Tetraspecific heterodimers targeting TNF-alpha, CD3, IL-5R, and IL-23R showed similar antigen affinities of the binding domains to corresponding single-chain variable fragments and single-chain diabodies. The TetraMab platform involves the combination of single-chain Fab and FV fragments in an IgG format. It also involves the knobs into holes technology. A TetraMab targeting HER3, c-Met, HER1, and IGF-1R binds to the cognate antigens with affinities comparable to the parental monospecific antibodies. This example also showed improved tumor growth inhibition. The DuoBody VHH platform involves the incorporation of VHH with different specificities to the heavy chain C-terminal ends of a bispecific Fc-silent IgG. An example was 1 or 2 VHH against EGFR, IL-6R, or NKG2D, which were appended to a HER2/c-Met bispecific antibody. Promising results were shown for NK cell-mediated cytotoxicity in vitro. The Tetra-KE platform is based on the tri-KE format, where small molecules containing a single variable portion, VH and VL, of an antibody link to two variable portions from other antibodies of different specificities. An example is the simultaneous engaging of EpCAM and CD133 for targeting carcinoma cells and cancer stem cells, along with a CD16 engaging moiety and IL-15 for NK cell expansion. The ANKET4 platform is based on ANKET technology, which is an antibody-based NK cell engager therapeutic technology. It is built on mAb fragments to induce synthetic immunity. ANKET4 induced NK cell proliferation and accumulation at the tumor microenvironment, and it had a higher anti-tumor efficacy. And finally, we have the Tri-TECM platform, where a tetrafunctional T-cell engaging antibody with built-in risk mitigation of cytokine release syndrome It enabled the fusion of trispecific TCE to an anti-IL-6R single-chain variable fragment antibody and triggered efficiently T cell activation and cytotoxicity against specific cells. Various clinical trials for tetraspecific antibodies are already underway. For example, GNC-035 targets CD3, CD137, PD-L1, and RR1, and it is indicated for the treatment of locally advanced or metastatic breast cancer. It is currently recruiting for a phase 1 study. Similarly, GNC-038 and GNC-039 are both also recruiting for phase 1 studies and are going to treat non-Hodgkin lymphoma or acute lymphoblastic leukemia, or relapsed or refractory metastatic glioma or other solid tumors, respectively. Tetraspecific antibodies have broad therapeutic potential across various diseases, specifically oncology, where they can engage immune system to attack cancer cells. For immunology, they may modulate immune responses for autoimmune diseases or allergies, and for neurology, they can target protein-protein interactions for neurodegenerative diseases. For infectious diseases, tetraspecific antibodies may also neutralize pathogens or target infected cells. The challenges of producing tetraspecific antibodies include manufacturing complexity. Efficient and cost-effective production methods are crucial for this. There is also the challenge of pharmacokinetics and safety, as understanding the behavior and potential side effects of these novel molecules are essential. For target selection, identifying the most effective combination of targets for specific diseases also requires further research. In the future, we may focus on the development of novel formats and engineering strategies for even more sophisticated multispecific antibodies. Continued clinical trials are also needed to evaluate the efficacy and safety of multispecific antibodies in various diseases, and we can hope that advancements in manufacturing technologies can enable more cost-effective and scalable production methods. Researchers may also use machine learning-based tools, which are quickly advancing with computational protein design. Physics-based approaches, like the Rosetta Suite, have successfully created de novo protein folds, enzymes, membrane proteins, ion channels, and self-assembling nanocages. They have also contributed to proof-of-concept vaccines for HIV, RSV, and influenza. In antibody engineering, these methods have enhanced antibody affinity, developed novel chain pairing technologies, and optimized developability. The impact of machine learning can be seen with tools like AlphaFold and RosettaFold, which revolutionize protein structure prediction, enabling the generation of accurate models in minutes. These tools optimize protein function and drug properties, including antibody epitopes, stability, and immunogenicity, significantly reducing the need for empirical testing. However, multispecific antibodies still face challenges in design with combinatorial complexity due to the vast number of possible BB combinations. Empirical screening is still crucial for selecting optimal multispecific antibody formats, addressing functional activity, developability, and pharmacokinetics. The unique properties of multispecific antibodies, such as interconnection through flexible linkers, pose additional challenges. In the future, ML offers potential solutions to explore and optimize the expansive space of these formats. Companies like Generate Biomedicines, Alpha Bio, and Nabla Bio are integrating ML with experimental testing to discover new protein binders and improve monoclonal antibody developability. Generating comprehensive datasets is also essential for advancing these capabilities. Biointron focuses on antibody discovery, expression, and optimization. From gene sequence to purified antibodies, it takes just 2 weeks. Our high-throughput platform enables the rapid expression of recombinant antibodies in HEK293 or CHO cells. We guarantee the delivery of 100 micrograms to 100 grams of recombinant antibodies. Biointron is also a well-recognized leader in VHH antibody discovery and production. With our self-owned alpaca breeding farm, we offer a 1 alpaca for 1 project commitment and guarantee the delivery of over 20 unique binders, high diversity, and large capacity. We also provide various services such as bispecific antibody production, afucosylated antibodies, single B-cell screening, hybridoma sequencing, antibody optimization, recombinant protein expression, CHO-K1 stable cell line development, and much more. Thank you for watching, and comment with any questions you have below. Don't forget to like and subscribe for more.